sharing. I'm guessing it's going to start. Online? Yeah. Well, they can see it after. Okay, so this is a workshop that I'm going to go over aspects of how to organize your data into a tidy format. So normally, like a lot of the time, you get like data that's kind of messy. You guys are probably a little bit familiar with that, working with yeah. So I guess the first thing is if you go to the code is manuscript.github.io/code-alongs. You can see here there's a section that says notes and code from the session will be available here. So if you click that, I don't have a lot of the stuff up, but right now there's some resources that you can go to. And there's, in particular, there's a paper by Hadley Wycombe about tidy data and how important that is for like analyzing data, especially in R. Like how you structure the data is important. Can save you a lot of time in running analyses and plotting your data. And before we start, if you just first make a new project, it's a new project, existing directory, go to, since it's always a good idea to work in R using projects, RStudio in their projects. I'm going to create a new project inside this folder called Code-along. Choose and then create a new project. So then inside there's already, I made just a file already called tidy data. So if you want, you can create your own R script by doing Control-Shift-N to create a new file, and then save that file and name it, and then type it up as we go along with the code. So if you haven't done so already, install readr, which is a package that we'll be using today to like be able to import data.
So you make a new project called Code-along, and then you make the R script?
Yes. You better just, let me just fix this. So that the console is here. Okay, there we go. It's a little bit better. Okay, so install that package, readr. Let me know when you've like started installing it, because I think it can be very big.
I just installed it.
You just installed it and you're installing it right now? Okay. So in the meantime, I'm going to go over, this is in the simplest form what tidy data is. This is the cheat sheet that you can find on the website that's created by RStudio. It's really useful, definitely worth looking at. So a tidy data set is essentially something that has each row is an observation and each variable has its own column. So for us, like each observation would be an individual who's had stuff measured at a specific time. So like for us, like we have like the one individual measured, and then that's one row, and then that's the baseline visit, and then the next visit would be another individual, and then that's another row, and so on and so forth. In like other cases, like if it was like an animal, you'd measure, each row would be the individual animal that you've measured, and then each variable is like the value that you obtain, for example, like the weight of an animal. I don't know too much about how cell culture research works, but I'm assuming like each row would be like the dish that the cell culture would be in. Is that, have you worked with cells?
I worked with cells.
So would it be like each?
It'd be like you have like say like two different cell types.
Yeah.
And then each cell type has different conditions. So like, it's almost like a matrix of high glucose, and then with treatment, without treatment, or something like that, and then normal control with treatment, without treatment. So I guess it would be
Okay.
4 things.
So like,
Or if you have,
and how many dishes would you have per treatment? Would be just 1 dish per treatment, or?
well, you want replicates,
So you'd
like
be,
3.
okay, 3, okay. So then it would be like, for each treatment, you'd have 3 rows for each of the dishes, yeah, the duplicates. And this is really useful and kind of important because I should probably check this. Because this is really important, at least within R, in that if you have a data set that's structured in this fashion, it's much easier to analyze it later on, especially like plotting it or any type of analysis kind of has to be put into this format. Otherwise, it's kind of a little bit messy. So if you want a more in-depth, more academic look at what tidy data is, you can look at the article published in the Journal of Statistical Software by Hadley Wycombe, talking about tidy data. It goes into quite a bit of depth about it, and I'm not going to go over it entirely because you can easily read it yourself. But I'm going to go over some example messy data sets versus clean data sets. So this would be more of a messy data set in that you have the religion along the sides of what they are, and then you'd have the income values along the top as individual rows, as well as the frequencies within. In R, this would be a little bit of a pain to be able to analyze because, well, why do you think that's the case? Can you guys give like a guess as to why this is messy versus tidy?
it's hard to see if you want to select combinations. You're assessing it more as a row than a column. I feel like each cell is very fragmented. I don't know how to describe it, but like, rather than factor with different levels, it's just all separate from each other.
That's, yeah, one way of looking at it. Like, so each variable, so the idea of a tidy data set is that each variable has its own column. In this case, income has 1, 2, 3, 4, 5, 6 columns. And frequencies doesn't have any columns at all, since each of these are frequencies, right? So, or counts. So they're the values inside the income, which is kind of a little bit messy and a little bit challenging to deal with if you want to analyze it or plot it. So a better way would be to have religion, income, and then each of the groups as its separate row, and then counts or frequencies as another row. So here's another way of looking at it. So row, these are each of the rows, and then the values along the top. This would be a more cleaner format. So you have the value here and then the income along the side here, as shown here. Let's go to, there was another one I want to show. Here's another example that might come up in some labs, depending on what your data is. And sometimes you don't always have, like, let's say you have an ID, like for example, a mouse, and sorry, not a mouse, like a treatment or something like that. And then each of these rows would be maybe the mouse measure of like weight, for example. Sometimes you don't always have full complete data on all the mice. So like this, for example, you'd have missingness. So you'd have some rows that would be completely empty. It's kind of irritating that way, because then you'd have a bunch of missingness in one of the columns, in some of the rows. So a better way to structure this would be to convert it into this format, so that you'd have the date with the day. So if you see here, this is day 1, day 2, day 3, day 4, and all the way up to day 8. And this is a better way of structuring it. So you have the date, right, with the actual day, the full day that you measured it, and then the values along the side. And then so then that way you'd maximize the amount of space within the data without having rows that are completely empty. Okay, so I'm going to show some examples of our own. I'm going to show 4 example data sets. The first 2 I kind of made up, but are maybe a little bit reflective of some of the work that students do in this department. And then the last 2 are actual data sets that Gila in our lab measured in our serum samples in the PROMISE cohort. And they were completely raw, and so they were obviously quite messy. So the first thing is, so load up readr. Now readr comes with, there is a base function called, this little hashtag just means comment. So the base function is called read.csv. Now how read_csv is different is that it's faster, and it does a lot less pre-processing. So like read.csv does a lot of stuff underneath that can kind of throw you off, while read_csv is a lot better at getting it in as is. But then if there are problems, there's a function that you can use called problems, and then you can specify like the name. So for example, if we have here, well, first things first, before we go for that, let's just run this. If we run this, this is an example data set that I wrote up. You can get it at, the nice thing about read.csv is that you can use a URL pointing to like a website. So if you just type it, all of this inside read_csv, you can then take that data from the CODIS manuscript. And I'll let you just type it up. You got it? No, you don't? Okay. Okay. So you can see here that there's kind of like, this is something that maybe some people in animal models might encounter or might set up their data as. So like you have each of the diet conditions, then you'd have an exercise group and a control group. And then you'd have like bunch of these values that, in this case, let's pretend that there's some their cell count of liver or something like that. So each of these values can only be obtained once from an animal. Obviously, you can only take out the liver from an animal once, and then you obviously can't take out liver again from that individual, that animal. So each of these cells represents an animal, and so sometimes you have cases where you might not be able to extract the liver, something goes wrong or whatever, and so you have missingness there. And then you have the case where, like, you know, there's missingness in some of the columns because it's just called exercise or control. So it's hard to understand what exactly is going on with this particular data set because it's a little bit messy. Now, how do you guys think we would fix this up? So how many columns would you have? 3. So the first column would be diet. The second one would be, the third one would be what? Control?
That exercise and control would be both in the second one, and the values would be.
Exactly. So each row would then be an individual animal, So you'd only have one value per animal. Now the next one, so we'll go through and try to like actually tidy this up, but let's first check out the next one. So the next one is where you might have a case where you have a cohort of people and you want to have it's like a nested case control cohort where you have people already specified as cases or controls, and then you have cases. And so in this case, you'd have an ID with the case. So you'd have this is the individual and their weight and their, for instance, testosterone, and then you'd have their matched pair of another individual with their weight and their testosterone control. Now, how would this be tidied up?
Columns.
Less columns, yeah. How many columns do you think we'd have?
4, 5, 4, 4.
Why 4?
5. Hold on. ID, control, or you just put like ID, weight, and testosterone.
Control case? Yep, that's right. That would, or
No, it's 4.
so 4, but then you'd need something to be able to match since it's a case control. So maybe you'd create something called match ID. So you'd say like this would be match ID of 1, and that would correspond to ID of cases and controls of this case and this case.
So you would have one with subject ID, the match ID, and then weight and testosterone.
Yep, exactly. Okay, so let's tidy it up. So we can do read CSV, and then we can do write CSV. And then specify the file. So this would be tidy data 1.csv. If we run that, so what does write CSV do?
Exports it.
Yeah, it exports it. So read brings it into R, and write puts it onto a file. And so this is the file name that I've said. And so because we're in the code along project, it will be saved under the desktop code along file folder. So if we run that, now if we look at it, open up it in an Excel type format. Okay, so a better way of doing it would be changing it from, so this would be diet. Yep. Actually, let's try to wrangle it. No, let's just do it manually. We have diet, we have treatment, then we'd have liver cell count or something like that. Another important thing is to name your columns appropriately. Always have like a consistent way of naming your variables, because that makes it easier for other people who might get your data, and also for you maybe a couple of months down the line and you're like, what are these? So in this case, this might take a little bit of work, but we'll just like we'll just start typing up a little bit. Exercise, I'm just going to do the first diet. I'm just going to delete this. So this is a little bit manual. There is ways to be able to process this in R. And that is a session I won't be going over how to do that right now, but that is something that I will be going over at a later time. Okay, so now we can just delete all of this. And this would all be diet A. So that's more of a tidier format. Especially when you want to start using like dplyr, which is like a way of wrangling or managing your data. tidyr, which is another package that deals with cleaning up your data or reorganizing it. And then plotting this would be a lot easier than the other way. You could also, if you wanted, include like a mouse ID or something like that. So that would be mouse 1, 2, 3, and all the way down. Oops. There you go. Mouse. Change that. Okay.
So you're not doing diet B or C then?
What's that?
You're not doing diet B and C.
Yeah, it just will take too long. Now it's useful. It's something like this. This type of data is no, just the lab. So this type of data, when you enter it in yourself, it's easy to put it in this format. But if you put it in the previous format, it takes a little bit of work to get it back into a nicer, clean format. So the best thing to do for tidy data is to get it into this format from the beginning rather than later on. So that's what you do here in this case. If you want to read that back in,
Did you save that file?
yes, it did. Yeah. So if you want to read that back in, do read CSV, tidy data on CSV. And then now we can look at it like this. Nicer format there. So this arrow just puts it into an object called BS. I'm not going to go over this one, but I'm going to go over something that maybe some people might encounter. This is what Gila had to do. This is so you can see here, the nice thing about read underscore, so in this case, TSV is for tab separated. So CSV is for comma separated, and TSV is for tab separated. And that's because each of the values are separated by a tab. And so it reads it in that way. But you can see right away that it says, oh, there's, unlike read.csv, and there's another function that does the same thing for tab delimited files, is that this will say, oh, there's a problem. These are the problem cases right here. And then you can go in and see what's going on. So in this case, I already know what the problem is. So if you just copy this line and do read lines, I think it's right. Yeah, there we go. We do read lines and then do write Right, right lines. That's a function too. So right lines is a base function. It doesn't come with readr, the readr package. It what it essentially does is it just takes this file, this data, and then saves it as is to this new file here. Now read lines, all that read lines does is it reads in each of the lines of this file. So it doesn't do anything else. So each line you can see here corresponds to what is contained on each of the lines. And you'll see each of these /t, that stands for tab. That's a tab character. So if we do write lines.txt, write lines, and hopefully it saves it. Okay, let's try that again. Might be the internet. There we go. So now there's this new file. No, that's fine. You can see here, each of these little marks indicates a tab. So this is maybe what some people might encounter. Oftentimes, like machines spit out this type of data, and in this case, Gila, this was what was it again for what? Spectra reader. So each of these values represents like a wavelength of something.
Yeah.
Or, yeah. So this is really messy, like a really messy format for like, there would be no way that you could analyze this in R. It just would be a mess. But at least the first thing that you could do is, so you see here that there's one row that doesn't really provide too much information. This next row or line has 1, 2, 3, 4, and those are the corresponding, I guess, plate locations within the, what would that be called? Just the plate? Yeah, the plate column, and then each of these other lines are the plate rows. And there's also these two extra columns here, these tabs that don't really have anything. So if we wanted to, we could run this read.txt again, except in this case, what we want to do is identify. So if you see here, there's these hashtags, hash, and that indicates that it's a comment. So we can say that that a comment is a hash. And then we want to skip one line because that first row isn't really important to us. And then the number of columns to read from, rows to read from that point would be, let's see, how many rows are there per plate? Eight. So it'd be a total of 8, I think. 9 plus the header. Let's see if this works. Now we still have a lot of missing values here. So we got rid of the first problem. Which is, let's see if, hold on. We got rid of the first problem, if you remember above, where it's just a bunch of like plate, and then also this end here. So we got rid of that problem. Oh, and in fact, that should be 8. So here, it is empty. So that should be 8. There we go. Now, to get rid of those first 2 columns that we don't really need, we do -1 to -2. So that says I want to exclude the first row, the first column, and the second column. There we go. Now there's some stuff at the end there that we probably don't want. So we can probably say trim white space is true. No, that won't work. So anything past 12 would be, will this work? Yes.
Why are the first 2 columns negative now? -1 and negative?
That's a good question. So when you use these little like square brackets, you're saying I want to access the the columns of this data set. And so when you say minus, you're saying I want to take this data set or this data set, not including the first row and not including the second, not including the first column and not including the second column. And then you can do it again. So you say for this new object, I don't want to include the 13th and the 14th column because they're just missing this for whatever reason.
Is the colon the 13 to 14 or the 13 and 14? Like say if you put 12 colon 14,
that would be, so 12 colon 14 would be from 12 to 14, yeah. So yeah.
What if you want to just remove 12 and 14?
If you just want to see an example of what colon does, if you just do 1 colon 10, that'll do a sequence of 1, 2, 3, 4, 5, all the way up to 10. That's all that the colon does. Good question.
So if you want to remove like 10 and 14, what if you just -10 and then a different bracket -14? Can you put a comment? Or something like that?
Say that again.
Like if you want to remove the 10th column and the 14th column.
Yeah.
Well, how would you do that?
You'd have to do, another way of doing that would be this, -13, -14. That would run those. I guess I should mention that Control Enter will send this line to our console here. So that's a little bit of a tidier data set. Now, if we wanted to tidy it up even more, we could use, so let's put it up into an object called, sorry about that. So put it into an object called DS for dataset. Now we have the DS object. And if you get the package tidyr, so if you don't have it, you can do install packages tidyr. I already have it, so I'm not going to run it. You can do this, so tidyr, and then if you do if you add, before we start doing anything else, we have to add kind of like a plate ID too. So if you do DS, call a new column called row. So when you do this little $, it's just saying the row, the column name row, and because that doesn't exist in this data set, when you do it like this, it creates a new column called row. And so all I'll do is just do 1 to 8. Now if we look at it again, oops. Oh, there we go. Okay, that's fine. So it's now created when I printed it, because I loaded dplyr, tidyr, which makes when you look at the data, it kind of changes the format of it. So it tells you some descriptive detail about it. So it tells you that there's 8 rows with 13 columns, and then it tells you what each of the column types are. So in this case, this is an integer because it doesn't have like any decimal places, and this is called a double, which is just something that has decimal places. Now, if we want to be able to convert this into a tidy format, we can do gather ds for the data set, and then call column plate column, and what was it called again? Wavelength. I want to exclude, so in this case, I'm going to change this to plate row, just to be consistent. So run this again. So you should now have something that looks like this. I've just changed the plate row, so I've rerun this line and the lines 12 to 14, so that there's now a new column called plate row. So now if we go back to here, if we do plate row, what gather does is it'll take it from this wide format. See how there's like values all along the top. Each of these represents each of these values represents a wavelength, and then each of these columns represents a column from the plate. So gather will take this from a wide format and put it into a long format. So the reason why I call this plate column is because it'll create 2 new columns however I tell it to call it, if that makes sense. It'd be better if I just run this. So what gather does is it takes it from that long format, the wide format here, and puts it into a long format. So there's a plate row, plate column, and wavelength columns. And so you can see here, this is the name that I called it. I said I want it to be called plate column, and the plate column represents the names of the columns at the top here. And then wavelength represents all of the values contained within here. Now I say minus plate row because I don't want that to happen to this plate row column. I want plate row column to stay as is, and to be sort of representative of each, to be unique within this, not unique, but so that each, yeah, so it matches. So plate row 1 will continue. See, you can see here, plate column 1 goes all the way down, and then represents so you can see here plate row 1, 2, 3, 4, 5, 6, 7, 8, and that represents plate column 1, and then plate column 2, and so forth. If you want to have a better look at it, so we call it ds2 on that, and then we can do view ds2 and run that. You have a longer format for looking at it. So you can see here plate column 1, plate column 2, plate column 3. So kind of all gather does is it stacks each of the values on top of one another, all the way down to plate column 12. So we've now tidied up a very, what's the word? Messy data set. Now, if you want this to represent, because in this case, what was the wavelength representative of?
Oh, the amount of.
Right, so each of these represents like the amount of protein within a value. So each of these columns would represent an individual, right? Or each of the cells, each of the cells would represent protein of an individual. So in this case, now you can then be able to correspond the plate row with the plate column to an ID. And this you'd have to do as a separate step, as like a separate separate data file. So then the last one is this, something again that Sheila did. And it's kind of a lot of missiness, a lot of messiness. So let's do the same thing that we did last time. Read lines, write lines, and we'll call it tidy data dot text. Let's just call it. Oh no, sorry, CSV. Sorry, CSV. Run that. Give me a second. Got it? Good. Do it. Yep. Yeah. Actually, in this case, we probably could have done write CSV. It would have probably done the same thing. But oh well, that's okay. Oh, no, it wouldn't have worked. There's too many problems, I think. Data frame, not true. Okay. Keep it like that. So close this. So if we look at this new, so you can see this is the problem, is this file contains a bunch of, this again came from the machine, straight from the machine, so it's very raw, and it adds some extra information to it. So like, for example, when it was created, and some other information that might be useful to somebody at some point in time, but for analyzing it, it's not really that useful. So we can, again, do the same thing as we did last time. We can skip up to line 22. So if we go here, we do copy and paste this line again, and instead of doing read lines, do read CSV. These. And then this time we do skip equals 22. Should work. So 22. There we go. So now we have a more clean data set. You can see this in adiponectin. Now, it's got a lot of stuff that we probably don't really need, and there's some like additional processing that we'll have to do before it's actually analyzable. But at least this way, it's a little bit cleaner that you can then eventually put into an analyzable format. Other than that, any questions?
If you want to delete like a row, it showed how to do it with a column.
Good question. If you wanted to, so I initially did it like this. So like 1 to 2, that selects columns 1 to 2. The other way to do it is when you don't have a comma, so you can include it in with a comma. When you don't have a comma, it defaults to selecting the column. When you include a comma, it defaults to selecting the column number and the row number. So in this case, hi. How's it going?
Good morning.
Good.
You guys are finishing up?
Yeah, we're almost finishing. That's okay. No problem. No problem. You can stick around to see Wendy do her code review.
Oh.
So this is this represents the row number. So like if I wanted to only select 1 to 20 rows and only select 3 to let's say 7 columns, that's how you select only those.
So then you just want to do column view 1 to 20 and comments, or just the rows?
Yeah. Or you just do like comma, and then that does selects only the first 20 rows and all of the columns. And that's it.
Thanks, I learned so much.
Yeah. And then now you can do your code review.
Yeah.